Hi, my name is Parag Maswani, and I'm the Chief Commercial Officer for Axovant Gene Therapies. On behalf of all my colleagues here, it's a pleasure to speak to you today to provide an update on the company and our pipeline of genetic medicines. These are our disclosures, and as a reminder, I'll be making forward-looking statements as part of this presentation. So a bit of background. I joined Axovant back in late 2018 as the company was well on its way towards building a presence in the CNS gene therapy space. We take a lot of pride in the progress that we've made in a very short period of time and have equally ambitious goals. We aspire to be the leading company targeting neurodegenerative diseases with clinical stage gene therapy programs. How do we intend to do that? Through a mix of a robust platform, through deep partnerships with experienced gene therapy organizations, and through our people. First, on platform, we are platform agnostic. We use both AAV and lentiviral vectors that are tailored to the unique needs of each disease. And we fundamentally believe that we have the potential in our programs to be either first in class or best in class, and we're amassing a data set to substantiate that. Partnerships. We're partnered with leading gene therapy organizations, including the NIH and the University of Massachusetts, who have done pioneering work in gene therapy research over the years, to organizations like Oxford Biomedica and ViralGen, who have built a capability and a competency in AAV lentiviral manufacturing that we believe is best in class. And finally, our people. We are 45 strong, based in offices in New York City and in North Carolina, and are individuals who have deep gene therapy expertise or deep knowledge of neurodegenerative diseases. But I think most importantly, what I'm proud of the people that we have here is the focus that we have as an organization to meet the needs of the communities that we serve in Parkinson's disease, as well as in the various rare diseases where we have programs. And so these are our clinical stage gene therapy programs. We have Axovant PD. This is our Parkinson's gene therapy program, where we deliver the 3 genes involved in the endogenous production of dopamine. This program is in a phase 2 dose escalation clinical trial called the Sunrise PD study. And earlier in October, we announced positive 6-month follow-up data from our most recent dose cohort. This is called cohort 2. The headline here, what we found is that clinical data across 5 distinct dose cohorts amassing 21 different patients, continues to demonstrate a dose-dependent efficacy and a favorable tolerability profile. And I'll share with you some of that data in just a second. The next step for the program and the path forward is to initiate and dose the first patient in Explore PD. This is a randomized sham-controlled clinical trial that we intend to start in 2021. We also have 2 rare fatal pediatric disease programs. These are in GM1 Gangliosidosis and a related condition called Tay-Sachs-Stanoff disease. Our program in GM1 is AXO-AAV GM1, and this is an AAV9-based gene therapy program that is in an ongoing clinical trial at the NIH. And we announced earlier this year that our current cohort of children with GM1 Gangliosidosis, 5 kids, are fully dosed and enrolled in the clinical trial, and we expect to have safety and efficacy data available from this cohort in the 4th quarter of 2020. In Tay-Sachs-Stanoff disease, an equally devastating condition, we're working hard towards the clearance of an IND, which we expect in calendar year 2020. I'll give you a bit more insight about each of these programs in just a moment. Let me briefly touch upon this notion of partnerships, and we're really proud of the partnerships that we've built, both in academia as well as on the manufacturing side. We know in the gene therapy landscape that manufacturing has the potential to be make or break and to be a bottleneck. We outlined that issue early on and established partnerships. On our Lenti program, we're partnered with Oxford Biomedica, who's a world-leading clinical and commercial manufacturer of lentiviral gene therapies, and we're working alongside them to support supply for our clinical studies and for eventual commercialization in the future. For our AAV programs, we are partnered with Andelin Biosciences, part of Nationwide Children's Hospital, and ViralGen, which is a leading CDMO that's been built as a joint venture between AskBio and Columbus Venture Partners in Spain. So let's talk about Axovant PD, or gene therapy for Parkinson's disease. And As I mentioned earlier, we're continuing to advance this program and generating data across multiple dose cohorts. We know the issue in Parkinson's disease. This is a big condition, over 1 million patients in the US, a multiple of that across the globe. And the challenge is, how do you maintain control of this disease and manage the bradykinesia, the rigidity, the tremors that have such a massive impact on people living with Parkinson's disease, where we know that over 50% of people with PD will develop motor problems after just 5 years, despite standard of care. And that standard of care in levodopa is 50 years old now. And we know that while it is effective, earlier in the disease in particular, it wanes in efficacy over time and is associated with treatment emergent side effects like dyskinesias. So we're trying to solve for that. We're trying to offer another approach where there is an opportunity to improve motor function and impact quality of life. And the way that we do that with Axovant PD is to use the lentiviral vector system with its larger packaging capacity to deliver the 3 genes involved in the endogenous production of dopamine. We're building on a body of evidence that goes back many years that integrates robust preclinical and clinical data from a first-generation program called ProSavin, and we're leveraging that with the data we've generated so far with Axovant PD, which has shown a dose-dependent improvement in the gold standard assessment of motor function, which is the UPDRS Part 3 OFF score. So we're making great progress here, and I'll show you some of the data in just a second. But before I do, I believe that a picture is worth 1,000 words. And what you see here are some videos, and these are videos of a patient in our clinical trial who has demonstrated a 22-point improvement from baseline in UPDRS Part 3 motor function. This is done in the off state. So this is done while they're washed off of background therapy. And in effect, it should give you a peer view. What's the residual effect of gene therapy on this patient? What you see very clearly in the upper right in the untreated patient is very characteristic symptoms of Parkinson's disease. So this inability to maintain voluntary control and this tremors. What you see 6 months later on the bottom in this video, which I assure you is played, is the very same patient, 6 months after receiving Axovant PD, washed off of all background therapy, and this is done in an in-hospital setting, and he's maintaining control. He's a profoundly different person. Now this is an N of 1, and it may not be representative of a full cohort, but it's an example of what a 20+ point improvement can do for a patient living with this condition. So as I mentioned before, how do we do it? We deliver the 3 enzymes required in the body's production of dopamine. We're trying to create dopamine biofactories. We do that by delivering these 3 enzymes that decatalyze this cascade to develop dopamine directly in the target tissue. We do that through a one-time administration of Axovant PD. And so this is our clinical program as it stands right now. We're building on the body of evidence with a first-generation program called ProSavin. And we're taking those learnings into the ongoing clinical trial. So we're in the Sunrise PD dose escalation study. This is in moderate to advanced Parkinson's disease, where we've shared data from cohort 1 in 2 patients. We've just shared data in our second dose cohort in 4 patients, and I'll show you some of that data in a moment. And the path forward now is to advance towards an expansion cohort versus a sham surgical procedure. This is what you see on the right part of this slide, and this is the Explora PD study, a randomized double-blind clinical trial where we would take the therapeutic, safe, and effective dose from the Sunrise PD study and advance that into the Explora PD study, where we expect to dose the first randomized patient in calendar year 2021. So what have we seen so far with Axovant PD, and why are we so excited about its potential and promise? This is a summary of data from both of our cohorts, cohort 1 and cohort 2, at 6 months. And what you had characterized on this slide here are 2 key assessments. The UPDRS OFF score, which is a measure of motor function, and the UPDRS Part 2 score, which is a measure of ADLs, the lived life. Are you able to bathe yourself? Are you able to brush your teeth? Are you able to feed yourself? It's a measure of real life and quality of life. And what we've seen here in cohort 1 is about a 65% improvement in the UPDRS Part 2 score of ADLs, and a 29% change from baseline in the Part 3 score assessing motor function. Now, if you compare that to what we've generated in cohort 2, we've seen a 71% improvement in the Part 2 score and a 21 point or 40% improvement in the Part 3 score. It's worth mentioning that we have 2 patients worth of data in this particular cohort. So we're comparing 2 patients in cohort 2 and 2 patients in cohort 1. Now, if you contrast that with what we've seen in the Hauser diary, we look for congruence. Do we see on this diary, which is a patient-reported outcome measure, congruence in outcomes with this clinician-assessed metric in the UPDRS score? So what you see here is an assessment of off time, which is the number of hours spent with Parkinsonian symptoms, and on time, which is the amount of time spent without significant motor abnormalities. And here across both parameters, we see clinically meaningful improvement, about 2.2 hours improvement in good on time, and a commensurate decrease of about 2.3 hours in diary off time. So encouraged with what we're seeing so far across these dose cohorts. And importantly, we continue to see clinical dose response in motor function across 5 distinct dose cohorts. So what you see plotted on this slide are average UPDRS Part 3 off scores and the improvement from baseline to 6 months, looking at the 3 different dose cohorts of first generation Pro-7, where you see about an 8 to 15 point improvement. And in orange, the cohorts 1 and cohort 2 for Axovant PD, where we see a 17 point and 21 point improvement respectively from baseline. And so, to bring this together and to cap my discussion on Axovant PD, we believe that we're building a potentially best-in-class product profile. We differentiate on mechanism from other gene therapies in development because we're delivering all 3 genes involved in the endogenous production of dopamine. We're hitting thresholds in terms of outcomes that are really deemed to be clinically meaningful as compared to what's been seen in other clinical trials and what's characterized as meaningful in the literature. And so we're advancing forward into our randomized clinical trial next year, where we believe we have the opportunity to develop a program that has the potential to meet the needs of an incredibly large total addressable market that you can see characterized here on this slide. Our base case view is there's roughly 200,000 patients that would be eligible for a gene therapy. These are patients who have moderate to advanced Parkinson's disease and are very characteristic of the type of patient that we're evaluating in our clinical trials. But we see the potential as being much more broader than that. And so we look forward to sharing more data from our Parkinson's program as we look ahead towards the next year and beyond. Let me move on to our programs in GM1, Tay-Sachs, and Sanoff disease. These are indications that we view to be very classical applications of gene replacement. These are diseases where there is an incredible amount of body of evidence that is known, and it makes them attractive targets for gene therapy. This is the characteristic biochemical cascade that leads to the manifestation of GM1 gangliosidosis, where you have a deficiency in the GLB1 gene, and GM2, where you have a deficiency in 2 genes, the HEX-A and the HEX-B gene. We are incredibly excited about these indications because we view them as high probability of success. There's an underlying neurobiology that supports the use of gene therapy for a long time, enzyme restoration and improvement in enzymatic activity. They're naturally occurring models of disease, felines and sheep who are born with these conditions just like humans are. And we pair those insights with clinically relevant biomarkers and validated measures of function. And we can achieve this through a single infusion of the vector and achieve broad distribution. So our first program is AXO-AAV-GM1 for GM1 gangliosidosis. This is a tough disease. It's almost universally fatal in the most severe forms of the disease in particular. And we know quite a bit about the natural history and natural progression of the disease, where you have this single gene defect that leads to meaningful morbidity and mortality. But I think most importantly, these kids diagnosed with this disease have no treatment options today. And so we aim to develop a program here through a one-time delivery of a functional copy of a gene delivered systemically with the AAV vector to improve outcomes, to restore neuromotor function and extend survival. And we believe we have the potential to do that with a vector that's proven to be effective in naturally occurring models of disease. I mentioned systemic delivery earlier, and IV delivery, and we really feel strongly that this is a differentiating feature of our gene therapy program. We know that in GM1 gangliosidosis, this is a CNS manifested disease, but there are multi-system manifestations that impact other organ systems throughout the body. So we know that when you have decreased enzymatic activity, you have the accumulation of toxic metabolites, and you can measure that in multiple organ systems throughout the body, in the brain, in the eye, in the heart. And importantly, we know through our interactions with key opinion leaders and understanding natural history that one of the #1 reasons why these children ultimately pass from this disease is due to respiratory insufficiency secondary to skeletal dysplasia. That's due to the accumulation of keratin sulfate. And so our goal here is through systemic delivery to have distribution of the transgene, not just within the CNS, in the brain and the spinal cord, but body-wide. And we've built some evidence for that through our partners and research that's been done at Auburn University, where there's a colony of naturally occurring GM1 felines. We've tested this approach, and what you can see in this video in the upper right is a very characteristic GM1 feline with difficulty with gait and mobility, hind limb weakness, and a very characteristic phenotype of a GM1 cat. What you see in the bottom right is another GM1 cat. This is a cat that's been treated with GM1 gene therapy, and what you find is a very different profile, a very different phenotype. Improved mobility and ambulation, a meaningfully improved hind limb strength that you wouldn't expect. But importantly, this cat's still alive out to 2+ years. This is in the setting of cats that are typically sacrificed at about 8 months. So we're really excited about the data that's been generated in these animal models that has shown the potential to restore wild-type survival in these cats. And this is with the very same dose, the very same route of administration that we've taken now into the clinic. And you can see some of this feline data here on this slide where it speaks for itself. You see in purple and in gray, essentially normalcy in these cats, whereas in green, you see what happens to the GM1 felines that have not been treated with gene therapy here. This is just an illustration briefly of the beta-gal enzymatic activity that can be achieved and has been observed across the CNS in these GM1 feline models. Here we're looking at cross sections of the brain and the spinal cord, and what we found and what the investigators have found is broad distribution of beta-gal staining, of enzymatic activity staining across the brain and the spinal cord with IV dosing of 1.5E13 and significant long-term coverage and expression. So where are we right now? We're in the clinic. We're the only GM1 gene therapy program that's in the clinic today. And as I mentioned at the beginning, we have fully enrolled our low-dose cohort of 5 children with type 2 or juvenile GM1, and we expect to share data across outcomes, safety, neuromotor function, development, exploratory markers of enzymatic activity, among others, later in the 4th quarter of this year. Importantly, we also have expanded our IND and our protocol to allow for a dosing of a 3-fold higher dose, so a 4.5E13 dose, and to treat and test this approach, not just in juveniles, but also in type 1 or infantile disease. And so we're looking forward to sharing additional data on this program in the 4th quarter of this year. And finally, let me talk briefly about our GM2 program targeting Tay-Sachs and Sanoff disease. Very similar disease to GM1 Gangliosidosis, where you have a fatal pediatric lysosomal storage disease that also has a well-characterized natural history that demonstrates developmental regression, blindness, epilepsy, all features that are well-characterized in the literature. But I think most importantly, no treatments available. We spend a lot of time with patient groups and patient organizations, and we're looking to advance forward a therapy that has the potential, again, through a one-time delivery of the 2 enzymes involved in Tay-Sachs and Sanoff disease, the HEX-A and HEX-B genes specifically, and deliver those directly to the target tissue in the CNS. And this is very important here for our GM2 program, where we're going direct to the target tissue through a dual route of administration, directly into the thalamus and into the spinal canal, which we believe has the potential for optimal transduction across the entire neuraxis. And importantly, we've seen some evidence of the benefit of this approach, where we reported the first evidence of disease stabilization in 2 children with Tay-Sachs disease as part of an expanded access study that's been shared and presented in the past at prior gene therapy research meetings. Similar to GM1, this is an illustration of the value of this combined route of administration, where the combined intraparenchymal and CSF dosing really is delivering strong outcomes here, where you see prolongation of survival in figure A, a maintenance and an improvement in the clinical rating score in figure B versus untreated cats, and neuracular distribution of the reconstituted enzyme. You can see that in the steep red staining across the brain and spinal cord cross sections with this combined route of administration. So where are we with this program now? Similar to GM1, this is the totality of our proposed clinical study design. We are expecting the clearance of our IND in calendar year 2020, and pending the clearance of the IND, we would intend to start a dose escalation series find the safe and effective dose, and move into a more efficacy-focused portion, here characterized as stage 2, to be able to advance this program to the next step. So I've given you a lot of information. Let me wrap up with this. This has been a year of transformation for Axovant Gene Therapies. We are laser focused on execution, clinical execution of our clinical programs in Parkinson's disease, as well as in the 2 rare diseases in GM1 and GM2 Gangliosidosis. We're continuing to focus on doing what's best on behalf of patients, and we are incredibly excited to share more updates and information with all of you in the months and in the years ahead. Thank you so much for your attention.